Good morning, everyone. Welcome to this week's work in progress. This week we have Eric Siegel. Eric is a PhD student in the Laboratory of Medical Zoology in the Microbiology Department at UMass Amherst. He graduated from UMass Amherst with a BS in Public Health and a BS in Veterinary Science, and then went on to the Royal Veterinary College in London and graduated with an MS in Veterinary Epidemiology and Public Health. Eric is also a contributing member to Nuvec's research team, and you will get to hear more about that work right now. Thank you for joining us, Eric.
Thanks for joining me today and taking interest in my work. I like repellency, I like drug design, I like population health, I like animal medicine, and so I put a lot of those together in the work that we're doing right now. So I'm going to tell you about one of the projects that we're working on. I'll start with a little bit of background to sort of set the stage for this work. So this work focuses on Lyme disease caused by Borrelia burgdorferi, which is transmitted by the black-legged tick in New England. The black-legged tick is the most frequent human-biting tick in the eastern United States. It has a very opportunistic method of host seeking, and they're everywhere. They have 3 life stages, a 2-year life cycle in New England, and they transmit the etiological agents for a number of human and companion animal diseases, including Borrelia burgdorferi, which causes Lyme disease, Anaplasma, which causes anaplasmosis, Babesia, which causes babesiosis, Powassan virus, and Borrelia miyamotoi. So I focus on Lyme disease with this work, which is the most common tick-borne disease in the United States and many parts of Europe. A hallmark symptom of Lyme disease is erythema migrans, which is the characteristic bullseye rash that appears after a tick bite. However, not all individuals develop or notice this rash. During the early stages, Borrelia burgdorferi disseminates through the bloodstream, which allowed spirochetes to invade multiple tissues and organs, and this rash is a manifestation of the spirochete dissemination away from the bite site. So if untreated, Borrelia can cause multiple body systems diseases, including disease of the musculoskeletal system with arthritis, neurological issues, cardiac issues, and psychological effects like depression and fatigue. And standard treatment for Lyme disease is doxycycline, with alternatives. However, Borrelia has remarkable adaptive mechanisms that make it difficult to fully eradicate. It can alter gene expression to survive in different environments, including the tick vector and various host tissues. And it can also enter a slow growth or dormant non-replicating state, particularly in immune privileged sites like the central nervous system and joints, which can contribute to post-Lyme treatment syndromes where symptoms develop despite antibiotic therapy. Cases of Lyme disease are underreported, but trends show that the incidence of disease increases with time and across space. I just show some CDC-produced maps here. And I also want to touch on the fact that feeding time is important, which I'll come back to a little later. There's a very good review article by Dr. Eisen in Ticks and Tick-borne Diseases that goes into good detail with this, but it's important to consider that pathogens require different tick feeding times for transmission, and this is based on differing mechanisms for each pathogen. So just to briefly summarize, ticks are a problem. We need new treatments for chronic and severe diseases. Feeding time is important for risk, and there's an important need to characterize new pathogens and develop integrated interventions for these diseases. So Borrelia is an obligate host-associated spirochete, and it is a causative agent of Lyme disease. It has a linear genome that's composed of a single linear chromosome and numerous linear and circular plasmids. Many of these are essential for host infection and immune evasion. Interestingly, it lacks common biosynthetic pathways, such as those that are needed for amino acid and fatty acid biosynthesis, making it really dependent on its host for nutrients. So Borrelia is motile, so it uses paraplasmic endoflagella located between its inner and outer membranes that give it this distinctive corkscrew-like motion, which also facilitates tissue penetration. And it's highly adaptable, so it can alter gene expression in response to environmental cues. These can be temperature, pH, and host immune pressure. And it utilizes this 2-component regulatory system, and this helps transition between the tick vector and the mammalian host environment. Important for treatment is that it can shift into these slow-growing or non-replicating states that contribute to antibiotic tolerance. So I briefly also want to introduce the enzootic maintenance of Borrelia in populations of Ixodes ticks. The cycle starts with uninfected larvae that emerge from an egg mass. Borrelia is not transmitted transovarially, so the larvae do not carry the pathogen. These uninfected larvae will acquire a host that is confident that is a competent reservoir for Borrelia. In the Northeast, these are mainly the white-footed mouse and the eastern chipmunk. And if these hosts are infected, then the tick will pick up the pathogen and molt into an infected nymph. This nymph could potentially feed on another competent reservoir host and pass the pathogen between them. And if the original nymph is not infected and the second host is infected, this is another opportunity for the tick to pick up the pathogen. The nymph will molt into an adult, and then the adult will feed on primarily a reproductive host. This is commonly the white-tailed deer, which is not a competent reservoir for Borrelia. And there are two opportunities for black-legged ticks to transmit Borrelia to humans or companion animals, and this occurs as either nymphs or adults mainly. And rough prevalence estimates are that there are about 25% of nymphs that are infected with Borrelia and about 50% for adults. And I just put these light bulbs here because we have opportunities for interventions at different steps in the cycle. And just as a couple examples, we have environmental-based interventions, so we can use habitat modifications like reducing leaf litter and the actual foliage that ticks can live in host seeking. We can also use chemical sprays and biological agents like acaricides and entomopathogenic organisms like metarhizium to directly target the ticks themselves in the environment. We can also use host-based interventions, so we can look at reservoir and reproductive hosts. We can reduce populations of them or feed them pesticides that feeding ticks will die from, or apply topical pesticides that will affect ticks when they walk on them or feed on them. And we can use things like tick tubes for mice or four-poster sites for deer. And lastly, I just want to bring up personal protection. That's the last line of defense against tick bites. And these include things like performing tick checks, wearing protective clothing with pesticides or light colors, using repellents, washing outdoor worn clothing and putting them in a hot dryer, and other methods. And today I just want to focus on an idea for reservoir host targeted interventions that focuses on Borrelia nucleotide biosynthesis. And I just have an example here of how nucleotide biosynthesis has been successfully targeted before, with some interest, but there were some issues with resistance development. So the example I give here is pyrimethamine, which has been used against toxoplasma and certain malaria parasites. This targets folate metabolism, but I focus more here on Borrelia's nucleotide biosynthesis. Borrelia has a restricted genome when compared to free-living bacteria, and this minimalism forces the salvage of essential nutrients from the host environment. And this is how they make up for the lack of biosynthetic pathways here. This case is de novo purine synthesis. When you consider mammalian cells, they produce purines mainly using de novo pathways when resources are abundant. A precursor in this pathway is inosine monophosphate, which is synthesized from other product pathways like the pentose phosphate pathway. IMP is eventually converted to AMP or GMP, and salvage is used in these cells to recycle free nucleotides or nucleobases, which can help conserve metabolic costs. And in the cases of cancer, they'll hyperactivate both ends of the pathway to accommodate high demand, especially the salvage pathway. Borrelia burgdorferi is dependent on the salvage of purine bases for nucleic acid biosynthesis. So Borrelia lacks the enzymes required for de novo synthesis of purines and is entirely dependent on the vector and mammalian host environments to salvage nucleobases, and this pathway of interconversion of purine bases to deoxynucleosides. So I have sort of an overwhelming pathway just to show what this looks like and based on my own interpretation of the literature. But I just want to show you what the basics of this looks like. So Borrelia brings these purine precursors into the membrane using these putative purine permeases. And it's through a series of interconversion steps that Borrelia takes these precursors and changes it into what they need in order to make DNA and RNA. There are key purine salvage enzymes, guaB and guaA here. This is an IMP dehydrogenase and GMP synthase that have been shown to be indispensable for Borrelia survival throughout the infectious cycle. And I focus on this pathway here that Borrelia relies on through guaB and guaA. This subpathway is important for the metabolism of hypoxanthine and adenine. And I just want to note that if there is enough free guanine in the environment, Borrelia can circumvent this pathway for survival, which this difference sort of infers how we can apply this to interventions and where we can. So my guiding research question is, how can we exploit the reliance of Borrelia on this pathway for therapeutics, some kind of measure of risk assessment, and ecological interventions? From the literature, we know that guaB exhibits an environment-dependent pattern of essentiality, so it is important in some cases and not important in others. There are three main environments that we work with in considering Borrelia. The first is in the mammal. So guaB is important for infection in the mammalian host. Normally, you can inject a mouse with cells and it will be infected. If you knock out guaB, the mouse does not infect it, so you absolutely do need it. Interestingly, in a tick that has fed, guaB only provides a fitness advantage in the vectored midgut. So if a tick has fed, spirochetes will still survive, but just with slightly less replication rates. And finally, if we cultivate cells in a culture from a guaB knockout, we don't see any defect in vivo with growth rates. So this shows that guaB is not needed in standard culture medium. So this is where our work has picked up. I just want to briefly discuss the product of an article that we published in October, which was titled Effects of guaB Inhibitors on Borrelia burgdorferi Growth in Standard and Modified Culture Conditions. I just show here the basic recipe for the standard growth medium for Borrelia, which is called BSK2. For our work, we grew cells in 3 culture conditions based on this medium. We had the standard formulation, a formulation that was serum-based, and then this was in an effort to mimic the mammalian environment as best we can. And then the final condition was just an exogenous addition of hypoxanthine, adenine, and the cofactor precursor for the guaB reaction, which is nicotinamide. And this was hypothesized that it could induce the essentiality of guaB in an environment where it is otherwise not needed. So with this work, we performed the first test of guaB inhibitors against Borrelia spirochetes to our knowledge so far. And we used 2 drugs at different concentrations. One is mycophenolic acid, which is an uncompetitive inhibitor of this enzyme, and 6-chloropurine riboside, which is a purine analog. Our results showed that the drugs were effective at reducing spirochete replication, but only in the modified growth conditions. So in standard growth conditions, the drugs did not have any effects, whereas once the cells hit log growth, they did not grow as much in the modified conditions that we set. So I just want to note that we do still need to do the experiments with the guaB knockouts. And I also have a note here, just considering why we wouldn't just use the purine spike with this modified medium, if that's likely the cue for guaB expression. And the answer is that we want the results to be a good indicator of what we might see in vivo, not necessarily where the drugs would be the most effective. And ultimately, the goal is to translate this into a substitute for a mouse model right now, since without a growth medium to test these drugs, we would need to use mice. And so if we can find something that would be comparable, then we can reduce the burden for needing mammalian models. So just to summarize so far, we have a crystal structure of guaB. There's this pattern of essentiality where guaB is needed sometimes and not needed in other times, which is likely due to the available amount of guanine in the environment. And we also show that possible guaB inhibitors show Borrelia static activity in cultures. So I want to make a brief couple notes about where this work could go and something that we're not really looking at right now. One of the goals is to develop a species-specific inhibitor as either a potential therapeutic or reservoir host treatment. And we would need to develop a species-specific drug since we do have an inosine monophosphate dehydrogenase, just like Borrelia. We can't have something that affects our own or that of mice. And since we understand the limited sequence identity between Borrelia's guaB and different mammalian equivalents, we can use a couple strategies to look for these drugs. There are 3 major classes of drugs that we can consider. These purine analogs, which our early work actually shows that Borrelia can take these in and phosphorylate them. This is appealing because there could be some kind of synergism with another one of the enzymes in this pathway that goes after guaB. We can also use direct binders of the IMP dehydrogenase subdomain, and the researchers who have characterized the crystal structure show that these do differ greatly from those of humans. And so that's where the opportunity for species-specific drugs comes in. We can also use these cation coordinating chelators. The needs for humans and needs for mice actually depend on different ones than humans, so that is another opportunity. And we've done a little bit of work with this. I do have a brief in silico drug design pathway detailed here that I follow. As of right now, I have 3 compounds that don't have names and would need to be synthesized by an organic chemist, but these may have some kind of species-specific effects and be non-toxic to the mammalian hosts. And I do have another note here that we would need more money for this, and we could also repeat this experiment with Babesia microti, which also has a guaB like Borrelia. So it's possible that we could find a drug that either affects both of them or a combination of drugs that could affect Borrelia and Babesia. So again, at this point, we aren't putting too much effort into this. The main focus of our work is now to optimize a culture screen and look at direct expression of guaB by Borrelia as it moves between its different environments. And so I'm just going to detail the small grant that we got from the European Society of Clinical Microbiology and Infectious Diseases to do this work. And I'll discuss the 2 aims that we have that we're working on going forward, which is more the work in progress part. So our first aim is to characterize, optimize, and use this culture screen that we started working on. And what we're going to do is take the serum-based assay that we had in our 2024 paper and grow cells at different concentrations, stratifying mainly by rabbit serum. And there's a quantitative PCR assay that's developed that we can use to actually characterize the expression of guaB from Borrelia grown in these different serum concentrations and see if we can get an assay that matches the expression that we see in the mammalian host that's infected with Borrelia. We could take this optimized assay and use biochemical methods, particularly LC-MS, to determine the concentration of the medium before and after log phase growth, mainly to understand how cells are utilizing the purines in the medium and how things are changing as cells are growing. And then finally, the end goal of this is to screen some possible inhibitors that may be a little bit more accessible. So if our in silico screen shows some compounds that could have effects that are that have names and are commercially available, we're trying to work on some more high throughput methods to be able to test those. And our second aim is to better understand the expression of guaB as a tick is feeding on the mammalian hosts. I'm just going to go back again here. These graphs right here show the expression of guaB after 2 and 4 weeks of mammalian infection, but nobody has characterized the expression of guaB as a tick has been feeding or in the first couple days of infection. So I have 2 hypotheses shown here in the bottom left about what this may look like. So the hypothesis is that guaB starts being expressed as cells are exposed to the mammalian environment or are exposed to some kind of cue that tells them that they're going to be in the mammalian environment. And we expect the expression of guaB relative to some kind of housekeeping gene to either start going up and continue through the first couple of days or reach some kind of peak after a certain point. And in order to determine this, we can feed ticks on a membrane every hour or 2, remove these ticks, and use this qPCR assay from the first aim to look at expression. And the goal consequence of this is that we may be able to use this as a more accurate and time-efficient substitute to physical measurements of ticks to indicate how long an infected tick has fed. And the goal of this is to develop a pipeline where we do a species test with PCR, determine if the tick is an Ixodes tick, to do the pathogen test to see if the tick is infected with Borrelia, and then finally to do the feeding time assay. So we can get individual and population level indicators of how long infected ticks had fed on hosts. And this could be a prototype for use in other pathogens. So while we don't understand the expression of the guaB equivalents in Babesia, for example, there are other genes that should have similar expression patterns to guaB in these organisms. And it's possible that we could develop this into a pipeline where, like I was saying, once you do a pathogen-specific qPCR assay, you then move on to the indicator of feeding time that is specific to that pathogen. So just to summarize what we've done so far and where we're going, Borrelia has this unique essential pathway of purine salvage. An enzyme in this pathway, guaB, which is inosine monophosphate dehydrogenase, is potentially a selective drug target that we could use for this. We showed evidence that cells can transport guaB inhibitors across the membrane and purine analogs, also to phosphorylate them. We showed that uncompetitive guaB inhibitors and purine analogs can reduce the growth of Borrelia at low concentrations. We proposed a modified growth medium that's hypothesized to make guaB essential, which is similar to what we see in the mammalian host. And then our two future perspectives are to develop some kind of therapeutic, optimize an assay to test these therapeutics, and hopefully apply these to reservoir hosts, either as a feed bait or broadcast spray. And then finally, the longer stretch is to see if we can use this as a high throughput molecular indicator of molecular feeding time, likely in some kind of pathogen testing program for ticks that are human biting. And so just to acknowledge all of the wonderful people who are helping with this work, of course, everyone in the lab, in particular, Connor Rich and Sanchana here, who helped me conceptualize and do the actual work for our original experiment, and some of the undergrads who are doing some of the current work for this work. This is now funded by ESCMID, so I acknowledge them, and NUVEC is also providing some funding for this. And I also acknowledge the individuals who helped me write letters for this grant and are contributing as good minds to consult for this work. And so I'm happy to take questions on what's been done, where this is going, or some kind of the foundations.
The feeding time assay is super interesting. And I'm just thinking about how we can use that at a population level to see like geographic variation in terms of maybe when people report, or just maybe just to talk a little bit more about how we could potentially use that.
Yeah, so the two ways that I'm thinking are, one, it could be reported to the individual for how long that tick has fed, or to their physician to make a determination of whether or not that's a risky tick, with a little bit more precision, as opposed to either saying with less granularity, engorged versus not engorged. And then the other way could also be to collect more information on how long these ticks are feeding. So right now, a lot of these indicators are self-reported, and if you look at the trends, the reporting is very underestimated by people. So people will underestimate how long a tick is fed on them based on their perceived risk. So they perceive that they picked up a tick when they were doing X activity, when it's very possible that they had either had that tick on them before or after. So this could be a way to gain more information on more granularity on how long pathogens could transmit to ticks, or to and from ticks, sorry.
That was a great talk. Thanks, Eric. That whole thing about, and kind of what Andrew was talking about, the figuring out how long it's been feeding, that's really cool. That could be really, that's really neat. I kind of had an unrelated question, and I can't even remember why I wanted to know. It was relevant to your talk, but I'll just ask it. Maybe I'll remember why I wanted to. Do you know the generation time of Borrelia, like in culture, like the doubling time? Do you by any chance know that?
Is Pat here? That's a better question for Pat.
Okay. But I had, it was connecting to something you said, and I think I've lost the thought now, but that might somehow connect to the guaB expression. And I lost my thought, but I guess now that I've asked the question, I'm still kind of, I'm sure we could just look that up, right? That's okay.
Bill, sorry, I was trying to unmute myself. It depends on the strain. So for like B31, a really common Borrelia strain, that's typically anywhere from like 12 to 24 hours. But we've had slower and fast growers before, but I think a 12 to 24 hour doubling time is pretty common.
What? So it would take that long?
Yeah, it grows pretty slow.
So then, but then it must be faster in, like when you get a bite, it can transmit the infection in, let's say, 48 hours. That would only be like 2 or 3 divisions.
Yeah, in log phase, we think it's like a 12-hour doubling time generally.
Oh, okay. Interesting. Yeah, cool. All right, thanks.
Hi, Dr. Mather, I see your hand is raised.
Hey, that was interesting. And so my question is how the thing that you're measuring, and I'm sorry, I'm not going to know any of the details, but you're going to measure something in your PCR, and it's going to give you some kind of number for how long the tick might have been feeding based on the concentration of it.
Yeah, so we can measure the amount of this gene produced that's relative to some kind of housekeeping gene. And the previous studies show that this number actually increases as a Borrelia has infected a host for a couple weeks. So the idea is that relative to this housekeeping gene, guaB should not be produced in high amounts, and then over time it should be produced more and more.
But is that going to be is that relative to the feeding time of the tick? Because that's what you're proposing that you're going to be able to measure, right?
Yeah, that's the hypothesis. But I don't know what that'll look like yet.
And I just wondered how much variability there is, because, now we can kind of look at a tick just based on sort of the scutal index. You have that picture, and we have a general sense of how long the tick has been attached. And I agree with you based on thousands and thousands of submissions to tick spotters that people are terrible about knowing how long a tick might have been attached. So being able to include that information and with some level of precision, the level of precision that we use is just, 1 to 2 days or 2 to 3 days or 3 to 4 days. And you still get occasionally people pushing back, thinking that whatever is transmitted as soon as the tick starts to bite. So I don't know how many people will believe that that's valuable necessarily, but some people seem to believe that it's valuable. But I'm more interested in how precise it is, and whether, what kind of range are you going to be able to measure over the course of the 4 or 5 days that the tick is attached.
Yeah, that's a great point. I'm kind of excited to see that, though. I don't know what it'll look like, but I think that's one of the fun things about it.
Tom, you might have been poking a question. I see Bill's coming back, but I just wanted to. So the clock that Eric's trying to set here is the clock based on the Borrelia cell cycle, which is maybe a proxy for feeding time. But that Borrelia cell cycle might be the one that's more relevant to infection, and that the feeding time is a proxy for the cell cycle as we've done it heretofore.
Yeah.
Does that make sense?
Yeah. Yeah. So but I'm wondering about, so you're looking at a measure of gene expression, which is still going to be related to the numbers of cells, right?
Yep. Which is probably somehow correlated with infectivity, right?
So you
Maybe.
I'm sorry to interrupt. As opposed to the number of cells, each individual cell should be producing more copies of this gene compared to other copies of genes that it would normally be producing. So it's a standardized measure to its other genes, not the absolute expression itself. I don't know if that's more useful or any more clear.
I don't know. For me, it's all a little esoteric. Thank you.
That's all right. Yeah. So you guys kind of got into this as well at the end there. Like, in addition to time that it's been feeding, and maybe I missed this, but could you also measure the level of gene expression? That might be a more direct measure of the infectivity or the pathogenicity, right? If it's upregulated, I don't know if there's like a clear on off, but if the guaB is upregulated, then things are brewing or something like, or maybe that's just the wrong gene for that kind of purpose. You know what I'm getting at? Like, instead of time of feeding, like just level of gene expression.
I think that's our main goal for this, though.
Oh, okay.
We're on the same page. Yeah.
Yeah. But like, instead of like, do you even need to know the time since feeding? Like, if the thing is being expressed, isn't that your measure of pathogenicity right there?
I think so, but there's always the possibility that instead of these graphs sort of kind of having this nice gradual slope that after 1 hour, you just see a peak, and that's as high of a peak as you're going to see. So the main thing to determine is, I guess, how does the slope change with time first? And then once
Okay.
you determine that, then you don't need to understand the feeding time. If you
Okay, I got you. Okay.
the curve to those.
I'm just remembering that back in the day, Joe Peisman had tried to quantify the number of spirochetes, I think, that were likely being delivered in ticks at any given stage of feeding. And I just what stuck in my head is that they may be injecting as few as a couple hundred spirochetes, and the variability then that we see in people becoming infected, it's sort of about an infectious dose, right? And also it's related to their own immunity. So everybody might be susceptible to different infectious doses. There may be some people that get infected very easily, and then other people that have a degree of resistance to infection for whatever reason. But I don't know, have you already sort of looked at the actual number of spirochetes that are, and tried to quantify them in ticks of different feeding stages?
No, our first step is to get membrane feeding to work. So
Ah,
we're a bit behind there. I think that's one of the first steps that we need to do, though. I agree with you.
and do you think that, let's say, mice and people will have different degrees of susceptibility to infection?
I don't know the answer to that. That's an immunology heavy question, right? Is that what you're referring to?
Yeah, I'm just wondering, because a lot of the experiments that we had worked on initially, all you have to test whether they get infected or not is with mice for the most part.
Yeah, I would imagine so.
Like the question sometimes comes up from people, and that is, they don't really understand the whole dose response, dose for infectious dose and everything. I guess they're starting to understand it a little bit better after COVID, because some people got infected and some people didn't, and it was related to their dose and exposure, etc. I just wondered if humans and mice have a different level of susceptibility to infection.
Yeah, I don't know the answer to that. I don't know if those experiments have been done before.
Probably not. I mean, it would all come back to, in this case, it would all come back to a precise measure of, let's say, the number of transcripts or whatever it is that you're measuring at any given time if you had the tick, right?
Yeah. So I guess we could actually do that if we took ticks that were submitted by people, ticks that were feeding on animals or feeding on some kind of mice, compare the two.
I guess one thing that we never know the outcome is whether someone, so people often will go ahead and take antibiotics, so that takes them out of the equation anyway. I just remember only one time that I can think of, that someone was bitten, and then they said later and they developed a Lyme disease rash.
Yeah.
And it was like, the question might have been, well, why didn't you take some kind of precaution? But getting that from a host of people, like some kind of a study, that would be hard to, I think most people would take most people will take the information that they were bitten by a black-legged tick and then take antibiotics, so that you would never know whether they got infected or not.
Mhm.
And then the people that end up getting infected, they mostly don't remember anything about their tick. They remember about it, they probably didn't take a picture or send it to Tick Report to be tested. Good for you that you're developing some kind of a standardized assay to evaluate all of that.
Thank you. Yeah. Hopefully we can figure out a way where it could be more useful than not.
I wonder if someone's degree of immunity is related to blood components. Then you could take different people's blood components in your assay and test it, right?
Yeah, that should work. Is there a specific component that you're thinking?
No, I mean, I have no idea. I'm sure some people know what are the elements of protection.
Yeah, I mean, I think one of the
Tick.
problems there is that human biological variability is usually as larger than the effects you're trying to measure.
Yeah, that's what I'm afraid of too.
Yeah, and people are trying to find correlates of protection for COVID vaccines for a couple of years, and humans are tricky.
It's been a great discussion, everyone. Well, if there are no more questions, we can leave the Zoom for today. And we'll see everybody in 2 weeks. Thank you, Eric, and thank you everyone for joining us.
Thank you, guys.